So multiplication of the viruses. After the attachment, virus need to enter inside the body of the host to multiply itself to use the host machinery. After the attachment, the nucleic acid components are released, as we have seen in the attachment. The nucleic acid components are released and they enter inside the body, inside a vacuole, and here the nucleic acid of the virus is active, but it is the temperature dependent. Temperature is very important. Also the other pH and the other, of course, they are the specific receptors which are attached. Therefore, it will be very easy for the virus to find it better environment to replicate itself. So how the virus will be entered inside the body? There are three mechanisms for the viruses to enter inside the body. First is the membrane fusion or hemifusion state. It is the process by which the cell's membrane is punctured and the virus goes through the membranes made to the further connect and unfolding the virus envelope. Then the endocytosis. Endocytosis is just like engulfing the food particle. The host takes place is virus particle through the process of endocytosis and essentially engulfing the virus just like it would be the food particle. The viral penetration, the viral capsid or genome may be injected in the host cell cytoplasm through the other ways, through the artificially, through the genetic, and through the other ways. So there are some other ways for the genetic inoculation. So we will see all of three these procedures one by one, that how the virus is capable of entering inside the body of the host. First of all, the virus entry through the membrane, which is called the semi-fusion and the fusion membrane process. In the viruses with an envelope, most of the times this membrane process happens in those viruses which have the envelope and they enter inside the body of the host through the membrane. Viral receptors attach to the receptors on the surface of the cell, just like we have seen in the last slides, that how the attachment with the virus receptors occurs with the host cells. The virus envelope fuses with the host cell membrane. Of course, after the attachment, the virus envelope will fuses with the host cell membrane and no bare virus enter into the cell. The virus only with the envelope will be capable, and then the virus envelope will be just capable with the cell membrane, and the bare virus is very easily capable of entering inside the body. The virus envelope blends with the host cell membrane and releasing its contact into the cell. And some of the classical examples for such kind of fusion membrane virus entry are the HIV and herpes simplex viruses, which enter inside the body of the host through this process. So here you can see the viral entry. Here is the membrane, here is the viral capsid which is involving it, and here are the receptors on the host. So viral capsid, this will be involved with this, and there is the membrane fusion. Here membrane through the nucleic acid, the capsid has captured the membrane of the host, and the nucleic acid is bare now, and nucleic acid can easily get entry inside the body of the virus. Now the viral entry via the endocytosis. Viruses, most of the viruses without the envelope, they are capable of entering inside the body through the endocytosis. Ingested by the host cell through the cell membrane, however, they are just like the engulfing the food particles, because virus will engulf the host will engulf those viruses. It just considering it just like body of their own, or they're considering it that these are the food particles coming from outside environment. They will be just at the door, just like knocking at the door, and cell will engulf the virus. Once inside the cell, the virus must break out of the vesicles by which it has taken up in order to gain access to the cytoplasm. Of course, to get the nucleic acid to multiply it need to get access to the cytoplasm. And the virus which are entered through the endocytosis inside the body, their examples include the poliovirus, hepatitis C virus, and FMDV virus. FMD stands for the foot and mouth disease virus. Here you can see that enveloped viruses, and so now here this is one very important phenomenon, that enveloped viruses are, most of them enter through the membrane fusion. However, some of the enveloped viruses are also capable with the endocytosis. Endocytome guarantees the low pH and exposure to the proteases which are needed to open the viral capsid and release the genetic material inside. Of course, because when the virus get entered inside the host through the endocytosis, The host is just considering them as their food particles and engulfing them. So it is also will be granted that there will be low pH and exposure of the proteases, so that virus get out and go inside the go release of the genetic material inside for the multiplication. Endosome transport the virus through the cells and ensure that no trace of virus is left on the surface. Of course, because it is engulfing just like the food particles, therefore, for the immune recognition, there will be no part left at the surface of the host. Now, through the genetic injection, attaching to the surface of the cell through the viral receptors, injecting only the genome of the virus into the cell. The rest of the virus particle, the rest of the capsid, will be outside the body, and this is restricted to virus in which only gene is required for the infection. Examples of the virus entry through the injection includes the bacteriophages. For example, we can take the example of T2 phage virus, which lands on the cell and its central sheath presses the cell membrane, and phage enters inside the body. So it is through just only the genetic material of the viruses entered inside the body of the host. It will be called genetic injection.